Welcome back to The Explainer. Today, we're going to tackle a gram-negative bacillus that's been showing up more and more in clinical labs. Leclercia Adecarboxylata.
So, let's start with the big question, right? Why are we suddenly seeing so much of this organism, which for the longest time we just thought of as an environmental bug?
Well, the answer, as is often the case in our field, comes down to technology. The widespread adoption of MALDI-TOF mass spectrometry has basically pulled back the curtain. It's overcome decades of misidentification, and it's finally revealing just how common Leclercia Adecarboxylata actually is.
Now, just so you know, everything we're covering today has been pulled together using a Gemini deep search of the current literature. If you want to dive deeper into the primary sources yourself, we've got you covered. The full list is available in the accompanying notebook document. Okay, here's our game plan for today. We'll start with a quick introduction to this emerging pathogen. Then we'll get into its clinical profile. After that, we'll hit the lab for identification, talk treatment, and finish up with the huge topic of resistance and control. All right, let's jump in. First up, the introduction. We really need to get to know this organism and understand how its history of being mistaken for something else has really shaped how we see it today.
You know, this timeline really tells a fascinating story. It was first described way back in 1962 as an Escherichia species. It wasn't until 1986 that DNA studies finally gave it its own home, the genus Leclercia. And this whole taxonomic journey is precisely why it flew under the radar for so many years.
So now let's look at how it behaves clinically. And what we find is this incredible split. It's really a tale of two different patient populations, and it all hinges on their immune status.
This slide just lays it out so clearly. On one side, in your immunocompromised patients, Leclercia Adecarboxylata acts as a lone wolf. It causes these severe monomicrobial infections like Bacteremia. But then on the other side, in healthy people, it almost always needs a crew. We see it in polymicrobial wound infections, usually after some kind of trauma and exposure to soil or water.
So the crucial takeaway here is this: a whole range of conditions that weaken the host's defenses or give the bacteria a way in, like catheters for dialysis or TPN, those are the major risk factors for these really invasive, serious infections.
Okay, time to bring it back to the bench. For those of us in the lab, accurate identification is absolutely everything, especially when you're dealing with an organism that has been a great pretender for so long. Here's what you need to be looking for.
So on your bench, the gram stain shows a pretty standard gram-negative rod. No surprises there. But it's the McConkey agar where the trouble starts. It's a lactose fermenter, giving you those pink colonies that look exactly like E. coli. And that right there, that mimicry, is the main reason for all the historical confusion.
Now, for anyone still using biochemicals, this table is absolutely critical. Your key differentiators are going to be a negative lysine decarboxylase and a positive melanate utilization. And here's another great clue. Leclercia decarboxylata is intrinsically resistant to phosphomycin, which really sets it apart from your typical wild-type Escherichia coli.
But, while biochemicals are still useful, let's be perfectly clear. MALDI-TOF mass spectrometry is the definitive method now. It's the tech that has completely unmasked this organism in modern labs, giving us that rapid, accurate ID we need. So, let's switch gears to treatment. Because there aren't any specific guidelines just for Leclercia adecarboxylata, our therapy has to be extrapolated from guidelines for other Enterobacterales. And the most important part, the absolute #1 rule, is that it has to be guided by antimicrobial susceptibility testing.
So if we look to the Dutch swab guidelines for Enterobacterales, we get a pretty clear playbook for empiric therapy. For sepsis, if you have a low risk of third-gen cephalosporin resistance, ceftriaxone is a solid pick. But if that risk is high, you need to shift to a carbapenem, like meropenem.
And for those resistant isolates, international guidelines get very specific. For a serious infection with an ESBL producer, carbapenems are your go-to. For carbapenem-resistant Enterobacteriales, well, it depends on the carbapenemase type. You're looking at newer combinations for KPC enzymes, and that ceftazidime ivibactam plus astrionum combo for the metallo-beta-lactamase producers. And a quick but really important point on pediatric patients. Case reports show that starting them on first-generation cephalosporins often fails. So just like with adults, therapy has to be targeted based on susceptibilities, with drugs like ceftriaxone or piperacillin tazobactam showing success. Okay, let's wrap up with the challenge that really defines so much of what we do: resistance. We're going to look at its intrinsic profile. the threats it can acquire, and how we can manage it from an infection control perspective. First, the basics of susceptibility testing. There are no species-specific breakpoints for Leclercia Adecarboxylata, so we have to apply the EU-CAST breakpoints that are established for the entire Enterobacteriales group. That's the standard we have to follow. Now this is the really concerning part. This bacterium is incredibly good at acquiring resistance genes. We've seen reports of isolates carrying genes for major ESBL families like CTXM, and even all the major classes of carbapenemases, NDM, VIM, and KPC. It's really alarming.
And that's what brings us to this quote, which really elevates its importance. Because it can be misidentified and because it can colonize patients silently, it can act as a stealth reservoir. It can quietly carry and then transfer these resistance plasmids to more well-known dangerous pathogens.
So infection prevention and control is just fundamental. It all starts with hand hygiene, of course, and applying standard precautions to every single patient. For those known multi-drug resistant strains, you got to escalate to contact precautions. And crucially, if you're dealing with an outbreak, you have to think beyond patient-to-patient spread and investigate things like contaminated medications or IV fluids.
And we'll end with this question. As our labs get better and better at identifying Leclercia Adecarboxylata, we have to confront its bigger, maybe more sinister potential. Is it just another opportunist, or is it actually a silent partner in the global spread of antibiotic resistance?
We really hope this briefing was valuable for you. Please subscribe for more explainers like this one, and we really want to hear from you. Share your own clinical insights and experiences with Leclercia Adecarboxylata down in the comments below. We're looking forward to hearing from you.